This video is about sexual reproduction at the cellular level and meiosis in particular. So with sexual reproduction, you always have two parents producing offspring, and the offspring have some genes from each parent, and more specifically, half of their DNA from each parent. Reproduction always requires the union of sex cells, which another scientific name for sex cells would be gametes. You're probably more familiar with the terms sperm and egg, which are, sperm are specifically the male gametes, and the female gametes are referred to as eggs, but the scientific name for egg singularly is ovum and plurally is ova. And sometimes students think, oh, you've made sperm, you've made egg, that's reproduction. But in order for reproduction to have occurred, you have to have fertilization. And that's when the sperm and the egg's nuclei unite. So it's not just enough for the sperm to penetrate the egg. The nucleus of the sperm has to fuse with the nucleus of the egg. And the new cell, that is the fertilized egg, is called a zygote. And that zygote will then start to divide mitotically to become more cells. We've already learned about mitosis in the previous unit. So now I want to talk about how are sperm and or eggs produced. And the name of the process is called meiosis. Be really careful, it's very easy to get meiosis and mitosis confused. And one silly mnemonic device that I've always used is mitosis happens in my toes, meiosis happens in my ovaries, which really only works exactly for half of us, but it's still a good mnemonic device anyway. So meiosis is the production of gametes, which is the general term, remember, for sperm and egg. And meiosis always reduces the chromosome number by half the number in the normal body cells. And that is so that when fertilization takes place, you get the normal number of chromosomes back. Otherwise, every time you had fertilization, you would double the number of chromosomes each generation. So for humans, sperm and egg have 23 chromosomes each, and when fertilization occurs, we get 46 as our chromosome number back. Another term that you're going to hear me using a lot in this video is somatic cells. So while gametes are sex cells, any other cell in your body is referred to as a somatic cell, and soma means body in ancient Greek. Most scientific words either have Latin root or Greek roots, and this one has a Greek root. So chromosomes occur in homologous pairs. Homo means the same, logos means structure. And if you see photographs of the chromosomes, they do actually look very similar and they look alike. But in addition to being similar in structure, they actually have the same equivalent genes on them. So I usually use a metaphor of like pairs of shoes. You might have 23 pairs of shoes, and you have a left and a right shoe of each of those shoe types. So you might have a pair of Vans, you might have a pair of Chucks, you might have a pair of Nikes, but they each, the Nikes look different than the Vans and the Chucks. So everything comes in pairs. Now in terms of how are they alike genetically, they carry genes for the same traits, although they might not be exactly identical. And it's best to use an example when I explain this. For example, if you have a chromosome from your mom that has a gene for eye color and a chromosome from your dad that has a gene for eye color, the gene from one parent might be a gene for blue eyes and the gene from the other parent might be a gene for brown eyes, but they're still both genes for eye color. So this cell is what we call a diploid cell. Some people pronounce it diploid, I pronounce it diploid. It really depends on, your teacher. But in humans, the diploid number or diploid number is 46 chromosomes, and it's the normal number of chromosomes in the body cells. Now, in this diagram to the right here, there's only 4 chromosomes, because if we tried to show you a picture with 46 chromosomes, it would get very visually confusing. So whenever we're showing you diagrams with chromosomes in them, we always use a simplified version just to get the idea across. So if you look at this diagram, you'll see 2 different sizes in this picture and 2 different colors. The chromosomes that are the same size are homologous to each other. So there's a homologous pair and here is a homologous pair. The 2 different colors represent which parents they came from. You will always see 2 different colors in diagrams of meiosis because in sexually reproducing species, there's always 2 parents. You may see a variety of different numbers of homologous pairs depending upon how simple they're trying to make the diagram. But for example, if this is a sperm or an egg production in you, the red chromosomes might be the chromosomes that you inherited from your mother, and the blue chromosomes might be the chromosomes that you inherited from your father. Now, this next cell is referred to as haploid. It has half the number of chromosomes of the somatic cells. So in this diploid cell, there were 4 chromosomes, and in this haploid cell, there are 2. You won't always know when you're looking at a picture of a cell or a diagram of a cell what the normal number would be. So there's a trick to figuring out, is your cell diploid and therefore a somatic cell, or haploid and therefore a gamete? What you always want to look for is the presence of homologous pairs. So does each chromosome have a pair? Now this one is pretty obvious because they're right next to each other. In some diagrams, they might not be at metaphase, they might not be lined up. So you may have to look around the cell a lot just to see, is there a match for each chromosome? Here, if you notice, there's 1 of each size chromosome, and here there's 2 of each size chromosomes, and the prefix di means 2. So you have 2 of each type of chromosomes. Now, this cell that has half the number of chromosomes is referred to as haploid. Now, what I like is haploid kind of sounds like half, right? It sounds like half-ploid. Don't spell it that way, but it does at least make it easier to remember. Now, some textbooks will use the term monoploid, and frankly, I wish more of them would, because mono and di are prefixes that you're familiar with if you've ever taken any, done a unit on biochemistry. You know the prefix mono means 1, and the prefix di means 2. Unfortunately, though, haploid is more commonly used. All somatic cells are diploid, I guess with the exception of adult red blood cells that don't have any DNA. And all gametes are haploid. Now, you're going to be doing some activities that are going to ask you, is a cell somatic? Is it haploid? Is it diploid? Is it a gamete? And I've seen students write somatic and gamete, or diploid and gamete, and those two don't work together. If it is somatic, it is diploid. If it is gamete, it is haploid. So don't get those confused. All somatic cells are diploid, all gametes are haploid. Now, there is a mathematical version of representing this, and it's 2n for the diploid and n for the haploid. So whatever your haploid number is, you'd multiply that by 2. So in humans, there are 23 chromosomes in a sperm or an egg, and that means 2 times 23, 46 in the body cells. Now, you might be wondering, well, why didn't they use X or Y like typically is used in algebra? And that's because there are X and Y chromosomes that determine whether you are male or female, and so it would have been very confusing if they had said 2X and X, because 2X means you're a female, and so it already has another meaning in this particular unit. So, the letter N is used instead of the letter X. So you probably don't have this picture in your note packet. However, what I love about this picture is that it really shows the difference between haploid and diploid. So here you have 1 of each type of chromosome. Now you have 2 of each type of chromosome. They are the same in size, they are the same in structure. If you look at the location of the centromeres, their location is the same. If you look at the banding pattern, they're the same. And when they stain, any areas that have a lot of adenines and thymines stain darker, and that's why you have those banding pattern. Down here you see 2 X chromosomes, so this individual would be female because there are 2 X chromosomes. If there were 1 X and 1 Y, it would be male, and the Y chromosome is much shorter. We will come back to that some more in the genetics unit later. But it's very important that each gamete have 1 of each type of cell. I don't mean each type of cell, I mean 1 of each type of chromosome. Because it's not just important that gametes in humans have 23 chromosomes, it's important that they have the right chromosomes, 1 of each type of chromosome, so that they have 1 copy of each gene, so that when fertilization takes place, you end up with 2 copies of each genes. It would be pretty disastrous if you were missing genes or had extra copies of genes. This picture is what we call a karyotype, and of course, chromosomes don't line up in this nice organized fashion in a cell, but scientists take pictures of cells and then reorganize the chromosomes if they're looking for chromosomal abnormalities. They would be looking for chromosomal abnormalities if a woman is pregnant and usually past the age of 35, or if they see something concerning on an ultrasound. And they remove fluid from around the baby, and there's always lots of cells undergoing mitosis in a fetus, because that's when we grow the fastest, and that's when cell division is happening the fastest. So it's really not hard to find a cell undergoing mitosis in the amniotic sac, in the fluid right around an infant that's developing. Just like it's very easy to find cells undergoing mitosis in a root tip, because they're constantly growing to find water. So this next diagram that I'm going to show you, I would like it noted for the record that there's a lot of stages of meiosis missing from this, because the purpose of this slide is to show you the difference between the two halves of meiosis. Just like when we were studying some previous units where you can really kind of distinctly split a process in half, there are two halves of meiosis. Now, the stages of meiosis are pretty much the same as the stages of mitosis, and their names are the same. So there's prophase 1, metaphase 1, anaphase 1, telophase 1, and then it starts over, and there's prophase 2, metaphase 2, anaphase 2, and telophase 2. And they're very similar to what happens in mitosis, but there are some distinct differences, and all of those differences happen in the first division. So this first division, and of course we're showing you metaphase 1, if you look, there are 4 chromosomes. There are 2 homologous pairs. So the homologous pairs line up and they separate into 2 different cells. So the spindle fibers would pull these double-stranded chromosomes apart. And now after the first division, you've gone from 4 chromosomes to 2 chromosomes. Now notice they're still double-stranded. One thing students often get confused about is they think double-stranded means diploid. This cell will be diploid, this cell will be haploid, because we've gone from 4 chromosomes and each of these chromosomes had a match. There are homologous pairs present. Now you have 2 chromosomes, but they're not the same size as each other, and they're not homologous to each other. So it's quite obvious that you have a haploid cell. Even if you didn't see this cell and know that the normal diploid number was 4 and this haploid number was 2, if you notice that these are different in size and shape, you know they're not homologous. No homologous chromosomes means you have a haploid cell, and definitely not a diploid cell. All of the really unique stuff that happens in meiosis happens in the first division, and I'm going to show you a little bit more of that in a future slide in a minute or two. But in the second division, we have the sister chromatids separating, and the chromosomes go from double-stranded to single-stranded. So the diploid to haploid happens in the first division, double-stranded to single-stranded is in the second division, and that's a lot like mitosis. So in that sense, metaphase 2, anaphase 2, it's going to look exactly like mitosis, only there will be half the number of chromosomes that there normally would be in that species dividing mitotically. Now, these cells are considered gametes. These are not quite gametes yet. So you cannot call these early cells gametes. In a male, this would be the primary spermatocyte. In a female, it would be the primary oocyte. But here, we're just really looking at the nucleus of each of these cells. So this is going to be an animation showing you mitosis. If you notice, you can tell instantly that this cell is diploid because you can see homologous pairs. This is homologous to this chromosome is homologous to this chromosome, and this chromosome is homologous to this chromosome. So in this imaginary species, the diploid number is 6. So in mitosis, chromosomes line up single file along the equator, and then the sister chromatids separate, and now we have 2 cells that each have 6 single-stranded chromosomes. We didn't have any change in chromosome number, and the cells went from diploid to diploid, or 2N to 2N, and you have 2 exactly identical cells and 1 division. With meiosis, if we still start off with the same species, if this was production of sperm or egg in that species, you'll see first that the homologous pairs line up. So it's not single file anymore. Each chromosome is lined up with its homologous pair. And you can't see this, but they find their homologous match during prophase 1. This is showing you metaphase 1. But synapsis is really, really, really important. And that's because you don't just want half the number of chromosomes. You have to have one of each type of chromosome so that you have one copy of each gene in the gamete. And that's so that when fertilization takes place, you'd end up with 2 copies of each gene. Otherwise, you could be, if you just divided them randomly, you could have the right number of chromosomes, but you could have extra copies of some genes and be missing copies of other genes, and you might not have the instructions to make all of the proteins that you need to make in the cell. So now, in the first division, it's already gone from diploid to haploid because we went from 6 chromosomes to 3 chromosomes, but notice they're still double-stranded here. Double-stranded does not mean diploid. Now, in the second division, that's when they go from double-stranded to single-stranded, and now we have 4 gametes with 3 single-stranded chromosomes each. Now you'll notice that you have different combinations of chromosomes. So the key with meiosis is that you get genetically different cells by the end of it. So we're going to talk about 3 sources of genetic diversity that are unique to sexually reproducing species. But please remember that any species can have mutations, and so mutations create genetic diversity regardless of whether you're talking about an asexually reproducing species or a sexually reproducing species. Now, consider though that in asexually reproducing species, they normally reproduce more rapidly. For example, bacteria divide approximately every 15 minutes. So more divisions means more chances for replication to go awry and to end up with more mutations. So in a sexually reproducing species like humans, there's a long time before we are actually even mature enough to produce gametes and produce offspring. And even so, we produce far fewer than a bacteria, which would divide and the population would grow exponentially. But in sexually reproducing species, we have 3 additional sources of variation in addition to the mutations that can happen in any species. And I'm going to list them first and then go into a little more detail. So the first one is random assortment of homologous chromosomes. The second one is crossing over. And the third one is random fertilization. Now we're going to do some activities that will bring home the point of random assortment and crossing over. Now, why do we care about variation? Well, variation isn't really necessarily important at the individual level, but at the species level, it's extremely important. Because if the environment changes and there's a great variation in the population, at least some of those individuals are likely to be able to survive long enough to produce offspring. So from the evolutionary perspective, it protects a species from possibly going extinct when there are environmental changes. So for example, if there is a disease that kills all organisms with a trait for green color, the population on the left, they're all green. So they would all die out. The population on the right, only the green ones would die out, and you might have some of the other variations that could survive. Now, of course, this scenario is not really a realistic scenario, but it gives you the basic idea. So with more genetic diversity, the population can better survive environmental pressures and less likely become extinct. So now let's talk about random assortment of homologous chromosomes. Now, everyone gets very excited about crossing over, but random assortment of homologous chromosomes is actually more important because it happens every time. Now, this is a dramatically simplified diagram compared to how this would be in humans, but it would be very visually confusing for us to have 23 pairs of chromosomes. But the lining up of the chromosomes along the equator during metaphase 1 is random. Now, not completely random because the homologous pairs still have to line up, but which chromosome ends up on which side can be random. So remember we said in an earlier slide, chromosomes of the same color came from the same parent. So if this is the process of meiosis happening in you, let's say that the red chromosomes are the chromosomes you inherited from your mom. The blue chromosomes are the chromosomes you inherited from your dad. So what messes with students' heads sometimes is they say, oh, well, how can there be male and female chromosomes in my gametes if I am male or I am female? But remember that when we talk about these chromosomes, these are the chromosomes that came from your parent. So by the time that these gametes get fertilized, they're really grandparent chromosomes. So if you look at this side, both of the red chromosomes lined up on the same side and the blue chromosomes lined up on the same side. And then when the cell divides and the spindle fibers pull chromosomes apart, then anything on the left side ends up in the left cell, everything on the right side ends up in the right cell. So if you look at these gametes have all blue chromosomes, these gametes have all red chromosomes. Now here, where the chromosomes have flipped and they're on the opposite side, you have one of each color chromosome in each gamete. So this would be representing male and female chromosomes. So there are 4 different combinations. These are identical to each other, but they're different than these two. Notice that here you have the long blue and the short red. Here you have the long red and the short blue. Now, there are only 4 possible combinations when the diploid number is 4 and the haploid number is 2, but what about in humans? So the way to figure out the total number of possible combinations of chromosomes in a gamete is a mathematical formula, and it's a simple one. It's 2 to the n number. So in humans, the n number, or the haploid number, is 23. So 2 to the 23rd is approximately 8 million possible combinations of chromosomes that can be in the sperm or egg of one parent. So that's quite a bit of variation. So the more chromosomes in a species, typically the more variation. Now, the next process I'm going to talk about is called crossing over, and it happens during synapsis. So remember I said a few minutes ago that synapsis is when the homologous chromosomes line up. And it's very important because otherwise you might end up with the wrong chromosomes in the wrong gametes. You want one of each kind of chromosome in the gametes. But during synapsis, what is formed is called a tetrad. So remember that each double-stranded chromosome has 2 sister chromatids. And remember that the sister chromatids are genetically identical to each other. But now, if you have homologous pairs, these are not genetically identical to those. Now, what this structure is called it's called a tetrad. Tetra means 4, and there are 4 sister chromatids. So when you have 2 double-stranded chromosomes pairing up, the name of the process is synapsis, but what you would call the cluster of chromosomes would be a tetrad because there's 1, 2, 3, 4 sister chromatids. Now, when those tetrads form during synapsis, sometimes a piece of chromosome will swap with the other equivalent piece of the other chromosome. So, for example, here you have some genes from the black chromosome that swapped with the genes from the white chromosome. And so now in the gametes, you can get a chromosome that has DNA from mostly one person, but a little piece of it from the other person. So if this is your dad, you now have a little piece of your mom's DNA where your dad's DNA was, and vice versa. And this creates new combinations of traits. It doesn't create new genes. The only place you can get a new gene completely is from mutation. But it's often referred to as genetic recombination because it's recombining different traits. If you have ever met a redhead that has no freckles, you do know for sure that somewhere in their family's history there has been a crossing over event because the gene for freckles and the gene for red hair are found on the same chromosome. Now, you may wonder, well, what about all those people that have freckles that don't have red hair? It is very possible and actually likely that they have a gene for red hair, but in order to get red hair, you have to be homozygous recessive for 2 different genes. And so you may have genes for red hair that are masked by genes for brown hair. So there are lots of people that have freckles that do not have red hair. So again, crossing over is when homologous pairs exchange segments. Now, if you ask people, oh, why is synapsis so important? Lots of students will say, oh, it's really important because that's when crossing over occurs. Now remember, crossing over occurs often enough for it to be really an important contributor of genetic variation. However, it's really important because it ensures that one of each type of chromosome ends up in the gamete. So there's really two reasons why synapsis is important. And the last source of genetic variation is called random fertilization. So millions of sperm are released, only one fertilizes the egg. And which sperm fertilizes the egg is random. Some of them will swim up the wrong fallopian tube, and they'll go against the current that's created by the cilia. There are these tiny little hairs that wave in the fallopian tubes that carry the egg down, and the sperm is swimming against the current. Some of them actually will end up having two tails, and they'll swim in circles, and they'll never reach the egg. It takes about 50 sperm to break down the nutrient coating around the egg. The heads of the sperm have enzymes in it that break them down, but once the first sperm actually penetrates the surface of the egg, instantly the chemistry of the surface of the egg changes and no more sperm can penetrate. But remember, fertilization doesn't happen until the nuclei fuse. Now, earlier we talked about all the different possible combinations of chromosomes that you can get due to random assortment of homologous chromosomes, but now you've got two parents. And if you have 8 million different combinations of chromosomes in each parent, if you want to know all the different possible combinations that each couple can produce, you have to take 8 million times 8 million. And that roughly ends up being about 64 trillion possible combinations of chromosomes. So we are all different, and the only time we have identical twins, and students often at this point in the lecture ask me, well, what about twins? Fraternal twins are actually when two eggs are released at the same time and both fertilized by different sperm. So you'll end up with siblings born at the same time. And they're not genetically more similar than regular siblings, although they do have the same maternal environment that they're developing in. So in that way, and they're born at the same time and their environment is very similar. So sometimes they seem more alike than regular siblings, but genetically speaking, they're not. With identical twins, you start off with one fertilized egg that quickly starts to mitotically divide, and you have a ball of cells, and then that ball of cells separates into two little balls of cells sometimes, and then you end up with two embryos and two fetuses and two babies. Now, this slide is all about what is the difference between the production of sperm and the production of eggs. First of all, I need to say that what happens in the nucleus is identical, but what happens with the cytoplasm is different. Now, first, before we begin, I want you to know that there should be 23 chromosomes in each of these sperm. There should be 23 chromosomes here, 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 and here. Now, I couldn't get them in there because if you've ever tried to make a text box, it ends up being bigger than the circle or the picture and ends up blocking it. So I couldn't get a text box small enough to write 23 in all of those places, but each one of these cells would have 23 chromosomes after the first cell, which is diploid. Because remember that the cell goes from diploid to haploid in the first division. So you're going to want to stop and write 23 in each of these cells. Now, I want to talk first about spermatogenesis, which is the production of sperm. And you can see the diagram of spermatogenesis on the left. The whole purpose when sperm is being produced is to make a lot of them because, if some of them swim up the wrong fallopian tube and it takes 50 of them to actually break down the nutrient coating of the egg, you need many of them. So sperm are small, or equally in size at least, and there's lots of them, and they have a tail, which means they are motile. They can swim on their own. The egg, you don't need as many of them. One is typically released each month. So what you really need is a big egg, because once that egg is fertilized, it's going to have to divide rapidly, and there's not a lot of time for the cell to grow in between each division, like typically happens with mitosis. And the type of cell division without growth in between is referred to as cleavage. And it really helps that work out well if you start off with one really big cell. So when the cells divide, the cytoplasm doesn't divide equally. So you get one really tiny cell and you get one big cell. These small cells are called polar bodies, and they really have one job, and that one job is to get rid of extra DNA and then just break down, leaving one good egg for each primary spermatocyte. I don't mean spermatocyte, I mean primary oocyte. This is a primary spermatocyte, primary oocyte. Cyte means cell, by the way, C-Y-T-E. So we have one big egg for every 4 small sperm. And then remember that the egg is non-motile. It will float downstream in the fallopian tubes, being moved by the cilia that creates a current. The cilia just sort of move. So now this last slide is just kind of a review of a comparison between mitosis and meiosis. And if you're asked during this unit to write a comparison, this would be a great place to start as a graphic organizer. So how many times does the cell divide in mitosis? Just once. How many times does a cell divide in meiosis? Twice. How many daughter cells do you get from mitosis? You get 2. And since meiosis has 2 divisions, divides in half into 2, and then each of those 2 divide in half to give you 4 cells total. Although, I guess technically, if you're talking about eggs, well, you get 4 cells, but you only get 1 viable, useful cell. Now, the parent cells in both mitosis and meiosis are always diploid. The daughter cells at the end of mitosis, still diploid. There is no change in chromosome number during mitotic cell division. At the end of meiosis, you have haploid cells that have half the normal chromosomes, so that when fertilization takes place, you have the normal number of chromosomes again. And then at the end of mitosis, the cells are genetically identical, and at the end of meiosis, the cells are all different because of random assortment of homologous chromosomes and crossing over. But remember, this is meiosis, so you can't consider random fertilization yet because it hasn't happened. At the end of mitosis, you have identical somatic body cells, and at the end of meiosis, you have gametes. They are sperm or eggs, depending upon if this is happening in a male or a female, and they have been produced in the testes or the ovaries, respectively.